Good evening, this is Mr. Wintermelon. So we have learned how does the plant makes its own food through photosynthesis. But how is the rate of photosynthesis affected? So in the last video of this chapter, we are going to learn about limiting factors of photosynthesis. So by the end of this lesson, you should be able to discuss how does changing the light intensity, carbon dioxide concentration, and temperature affect the rate of photosynthesis, as well as point out which factor is the limiting factor of photosynthesis at a given experiment. So we have gone through what is limiting factor in the chapter of enzyme, but let us just recap a bit. A limiting factor is a factor that directly alter or affect the rate of reaction if the supply is increased. In a chemical reaction, there are several factors that can affect the rate of reaction, and the factor with the lowest supply is normally the limiting factor. But to be honest, we are using the word supply very loosely down here because you can't say the supply of temperature. You can't really compare supply of temperature to the supply of substrate because energy and a physical molecules or the substrate, you can't really compare which one the supply is more, which one the supply is less. So for photosynthesis, the three external factors that affect the rate will be light intensity, concentration of carbon dioxide, as well as temperature. Temperature because photosynthesis is an enzyme catalyzed reaction. And a typical setup you will see is something that looks like this. You will have a water bath with an aquatic plant that's inside, and there will be sodium hydrogen carbonate solution. This carbonate solution will help to provide the carbon dioxide needed for photosynthesis. And to change the concentration of carbon dioxide, we just change the concentration of carbonate that is inside the solution. There will also be a lamp or a light source, and by changing the distance of the light source from the setup, we can change the light intensity. Temperature, we can just change it using a water bath. So first, let us take a look at the graph of the rate of photosynthesis against light intensity. And a typical graph will look like this. So first, there's a linear increase, and with increase in light intensity, the increase will actually slows down. That's why it will eventually plateau. Plateau means that with increase in light intensity, the reaction doesn't go any higher. That's why it's flat. So how do we make sense of this graph? During the linear increase and the increase at a decreasing rate part of the graph, all the factors are limiting factor. But of course, we can't really tell whether temperature is a limiting factor in this case because I did not state what is the temperature. So this might be 20 degrees Celsius and it is not the optimum temperature of the enzyme. So temperature will be a limiting factor. But let's say this graph is actually at 40 degrees Celsius, which is the optimum temperature of the enzyme, then temperature is not a limiting factor. So we can't really tell whether temperature is or is not, but since there's a carbon dioxide concentration here, we know that carbon dioxide concentration is a limiting factor, as well as light intensity. Because as we increase the light intensity, the rate of photosynthesis does go up. However, after the plateau, light intensity is no longer a limiting factor. Yeah, this phrase is actually very important. After the plateau, the factor that is increasing, or the x-axis, is no longer a limiting factor. Because when we increase light intensity, the rate of photosynthesis doesn't go up. We have to increase other factors such as carbon dioxide concentration to increase the rate of photosynthesis even further. So right here, carbon dioxide concentration is still a limiting factor, and maybe together with temperature. Next, we will take a look at the graph of increasing carbon dioxide concentration. Wow, looks familiar, right? So the graph of rate of photosynthesis against the rate of carbon dioxide concentration will actually look similar to that of light intensity. The only difference is after the plateau, instead of light intensity, it will be carbon dioxide concentration is no longer a limiting factor, because you can see that we increase carbon dioxide concentration, but rate of photosynthesis doesn't increase. Lastly, let us take a look at the graph of temperature and rate of photosynthesis. So how does the rate of photosynthesis changes with increasing temperature? Will be something like this, which should be very familiar to you because it is basically the enzyme activity against temperature graph. There's an exponential increase until the optimum temperature, after which there will be a sharp decrease because enzymes have been denatured. So this shouldn't be a surprise, and you should be familiar with the graph. At the optimum temperature, temperature will no longer be a limiting factor since a further increase in temperature will not cause the reaction to go even further. In fact, the rate of photosynthesis will decrease when we increase the temperature. So since temperature is no longer a limiting factor, other factors such as light intensity as well as carbon dioxide concentration is the limiting factor. At the part of the graph whereby there's an exponential increase, at temperature below the optimum temperature, carbon dioxide concentration, light intensity as well as temperature itself will be the limiting factor. So now that we understand how each factor affects the rate of photosynthesis, let us see how graph will look like when we combine the factors. We will first start off with light intensity as well as carbon dioxide concentration. So over here, I have the earlier graph of increasing light intensity at 0.03% carbon dioxide concentration. So look at what happened when I increase the carbon dioxide concentration to 0.13%. Same thing, at the plateau, light intensity will no longer be a limiting factor. But for the graph of 0.03% carbon dioxide concentration, at the part where the graph is increasing, the limiting factors will be light intensity as well as carbon dioxide concentration, because I know that at that point, when I increase the light intensity, the rate of reaction will go even higher. And when I increase carbon dioxide concentration, I can see that the rate of photosynthesis is also higher. However, if we look at the graph of 0.13% carbon dioxide, maybe I can only say that the light intensity is the limiting factor, because in this case, carbon dioxide concentration have already been increased. And the subject of carbon dioxide, right? Sometimes the carbon dioxide can be quite a sensitive thing, because like human cell, too much carbon dioxide might not be good for the plant. So if 0.13% is the so-called optimum carbon dioxide concentration, because we cannot have 100% carbon dioxide concentration, because the plant cell will just die at 100% carbon dioxide concentration, since no oxygen for respiration. So for the increasing part of the blue color graph, normally we will just say that light intensity is the limiting factor. But this is provided we have another graph for comparison. If it's just an individual graph, we can say that carbon dioxide concentration as well as light intensity is the limiting factor. But since we have two graphs for comparison, it is safe to just say that the light intensity is the limiting factor. Next, let us look at graph whereby we add in temperature as well. So what I have down here is I have decreased the temperature from 30 degrees Celsius to 20 degrees Celsius. If we compare the red dotted line and the red solid line, the limiting factor here is the temperature, because we can see that increasing the temperature increase the rate of photosynthesis. If we compare the red dotted line and the blue dotted line, the limiting factor at the red dotted line at that point is the carbon dioxide concentration, because when increasing the carbon dioxide concentration, the rate of photosynthesis actually increase. For comparison of these two dotted lines, we will normally not say that the temperature is a limiting factor since there's no change in temperature. We do not know whether 20 degrees Celsius is the optimum temperature of the enzyme or not. It is only when we compare the red colored dotted line and the blue solid line, then we can say that temperature as well as carbon dioxide concentration are the limiting factor here for the red colored dotted line. For the blue colored solid line, maybe carbon dioxide concentration as well as temperature is at the optimum level, so maybe they are not limiting the rate of photosynthesis anymore. We will not know this unless we do further experiment. But what we can be certain of is light intensity is no longer a limiting factor, because increasing light intensity will not increase the rate of photosynthesis even further. So to summarize everything in words, photosynthesis is influenced by three factors. At least for your O-level syllabus, it is three factors. They are light intensity, carbon dioxide concentration, as well as temperature. Why does light intensity affect the rate of photosynthesis? Is because they are responsible for the amount of chemical energy you have to convert carbon dioxide into glucose, as well as photolysis of water molecules. So at low light intensity is the limiting factor because light is needed to split water molecules into hydrogen ion, which is needed to reduce carbon dioxide. And light is also being converted into chemical energy so that the plant have energy to reduce carbon dioxide into glucose. At high light intensity, other factors such as carbon dioxide as well as temperature could be the limiting factor, since carbon dioxide is the substrate for photosynthesis. So carbon dioxide concentration affect the rate of photosynthesis is because it is the substrate. You need carbon dioxide in order for you to get glucose. The glucose is made up of carbon dioxide. So at low carbon dioxide concentration, carbon dioxide is the limiting factor since there is insufficient substrate. At high carbon dioxide concentration, it is no longer a limiting factor. Other factors such as light intensity or temperature could be the limiting factor. As for temperature, photosynthesis is catalyzed by enzyme, that's why it is sensitive to change in temperature. At low temperature, temperature is a limiting factor, and enzyme activity will generally double with every 10 degrees increase in the temperature until the optimum temperature. So you cannot just write as temperature increase, the rate of photosynthesis increase, full stop. You need to write as temperature increase, rate of photosynthesis increase until the optimum temperature. Because at optimum temperature, the temperature is no longer a limiting factor since any increase in the temperature will not cause the rate of reaction to become faster. So light intensity or carbon dioxide concentration might be the limiting factor in this case. And this will be all for the chapter of nutrition in plant.